speakers like to take a look at their audience, but it's up to you.
Maybe.
But anyhow, keep your keep everything on mute, just so there's no background noise to interrupt our speakers. And thank you everyone for coming tonight. I'm Gail Baker. I'm co-chair of the programs, along with Angela Soto, and we put all these
This meeting is being recorded
together for you. And we do get input from all our members, and this topic and these speakers were suggested at one of those meetings where we had people suggesting things. You can go to the next slide, Jenny. So I don't know how many of you have been lucky enough to have seen some of these parasitic plants that are that don't photosynthesize, but some of you at the coast, or if you've gone hiking at the coast, are familiar with ground cone, which used to be Bosniacia, the way I learned it, now is in a different genus, and also Orobanchaceae uniflora, also in a different genus. So our speakers are going to tell us about it. So these plants are really exciting for me, and hopefully you'll find them as exciting too, and really have a good search image in the field this spring. Okay, next slide. So I'm really happy to welcome two speakers tonight. These are two very enthusiastic experts on parasitic plants. They're specialists here. Our Adam is an assistant professor of biology. He's coming from a couple of time zones away. He's the curator of the herbarium at University of Wisconsin in LaCrosse, and he has been there since 2022. He has been studying the systematics of these parasitic plants and the parasitic plant lineages of quite a few of the different ones, including mistletoe and other genera and families. And Alison has served as a curator of the Center for Plant Diversity Herbarium at UC Davis. So she's in our zone since 2020. I'm going to ask people to mute if they can. I don't want to talk over people. Okay, so before Alison was at Davis, she served as an assistant rare plant botanist for California Native Plant Society, and for many years before that as a botanist at Yosemite National Park. And her graduate work was investigating the trophic switch from photosynthesis to parasitism, which I think is kind of exciting. So with that, I would like to welcome you both, and we'll turn it over to Adam's going to start, and Alison will follow up. Thanks again, you guys.
All right, good evening, everybody. Thank you, Gail, for that introduction, and of course, the invitation to speak with you all. This is really fun to get to talk about one of my favorite groups of plants, and to do it with Alison, who has been a great partner in all of this for the last 10 years for me, and she's been studying it longer. So I'm going to start out by giving a talk about a lot of the newest changes, taxonomic changes that are really clustered within a small group of the parasitic Orobanchaceae. And actually all of these species are found in Oregon, so that's kind of cool. And then Alison will take over and give a little bit bigger picture description of some of the major groups of these holoparasitic Orobanchaceae, and then also cover some of the other species that you're likely to, or at least potentially able to find when you're out on hikes, that maybe have had more stable taxonomy. So I'll just jump right in here and start with some of these recent changes. As you maybe saw from the original slide, or many of you are probably familiar with some of the older names for these taxa, or in the genus Orobanchaceae or Bosniakia, things like that, and might be curious as to why there's been so many changes and even what those changes are. I know it can be pretty hard to keep up. And so a lot of this kind of came from my dissertation research. I did my PhD at UC Berkeley from 2012 to 2017, and really was able to have the time to do a lot of these changes, to do the research required to update our classification. But of course, this isn't just my effort, but also the effort of many other people, as I'll try to allude to or give credit to throughout. So first of all, some of the recent changes have been because of changes in classification. And so, as biologists, we like to try to update our classifications when we learn more about the relationships of different species and genera and families. And so as a result of some of that evolutionary understanding through phylogenetics, we've had to change the names, right, of some of these plants so that we can keep, families, all the things in a particular family being each other's closest relatives, and same with the genus and species. So here are just a few examples of classification changes. So first of all of our native species in Orobanchaceae were moved to a genus called Aphyllon, which is actually, it's a name that was first proposed back in the 1800s by Asa Gray, kind of the first or certainly preeminent 19th century botanist who was trained in the United States and became a professor of botany at Harvard. He proposed that name, but then European taxonomists kind of lumped all of the North American species in with Orobanchaceae, and that's kind of how the taxonomy sat for almost 100 years. And so really this is an old name. It's a really beautiful name, I think, because it refers to the leafless aphyllon, meaning without leaves, the leafless nature of these plants, right? They're not photosynthesizing. So that's where that change came from. And also for similar reasons, one of the non-native pathogenic species here, Orobanchaceae ramosa, also had to be moved to a new genus called Holoparasitic. And again, it's just a new name for the same species. And then finally, this last one is a change of rank, and so Aphyllon uniflorum subspecies occidentale, which is I think how it was described in the Flora of North America or recognized, has now been upgraded to species rank through molecular phylogenetics and additional morphological study. It's clear that it's just as unique as any of the other Aphyllon species, and so used this earlier name Aphyllon purpureum. A second set of new changes has to do with just new discoveries to science, which is always exciting. As well studied as the botany of the Western United States has been, we're still making new discoveries and better understanding the biodiversity that's out there. And so this was a project that publication that Alison took the lead on and I contributed to, but actually results in kind of the culmination of almost 100 years of work and observations by other botanists by this unique species that I'll talk a little bit more about later in the talk. And by the way, there's a great Lane County connection here. It's thanks to Tanya for sharing some of her samples and observations and knowledge about the local populations of this particular species as we were preparing that publication. You guys are right at the northern range limit of this species. And then more recently, there was another species description, Aphyllon franciscanum, which was split from Aphyllon fasciculatum as a result of some both the phylogenetic work, but also a morphological project. And hopefully we'll have time, I can kind of explain to you some of the methods and kind of a behind the scenes of how we were able to figure out how to tell these two species apart. So why has there been so much change? Well, like I said, I spent a lot of time writing my dissertation in this particular group. And one of the things that came out of that was a recognition that these parasitic plants have such a reduced morphology, right? There's no leaves, there's no shoot architecture. They're only really visible when they're flowering, right? There's a lot of reasons why they maybe haven't been as closely studied as other types of plants, and this reduced morphology maybe obscures some of the biodiversity that exists in this lineage. And also, describing new species and doing this takes a lot of time. And like I said, it's not just me who's put this effort in here. Of course, Alison has done a lot of work, and I have a list here of several other collaborators on the research that I've talked about. And we've also built on earlier botanists who maybe hadn't formally described some of these species as new, but collected specimens, made observations of differences, all sorts of different types of evidence that we were then able to finally publish these species descriptions. Just as a quick introduction, though, to the family at large, many members of this family are partially photosynthetic, like the beautiful Castanea that is in your logo, right? So these are also members of the Orobanchaceae, but still are capable of photosynthesis, and will also sometimes supplement that with parasitism, root parasitism. Whereas you can see down below a couple of examples, right, of aphyllon or some of the Copsiopsis and other lineages that are completely non-photosynthetic. And all of these parasites form these root host or these host parasite connections, which you can see at the lower right. A really good example of that. The aphyllon in this case is the darker tissue behind, and this bright white root is actually the root of the host that is then being attached to and parasitized. And that's how the parasite gets all of its nutrition. With these completely non-photosynthetic parasites, they don't have any appreciable root system. You can see these little, just these little tiny projections, almost just look like, little worms coming off the base of the plant. That's all that's left of the root, and don't have really any nutrient acquisition function. Maybe help a little bit with support and keeping that host root close enough that the hostorium, the parasitism organ, can attach and keep siphoning off those resources. But again, a lot of morphological reduction. So you can see that in a couple of examples here of different plants. The one on the left is from a herbarium specimen, and the one on the right is from a collection I made in Florida. And again, you can see that connection, that intimate physical connection between the host and the parasite. And as I'll continue to talk about, if you're interested in identifying these species, the easiest way that you can do that is by figuring out what the host is. Obviously, you can't always dig up these plants, right? Maybe you're in a protected area, or you just don't want to dig them up. And so sometimes, you'll have to try to guess which, what host plants are maybe nearby. Maybe you have a list of some potential host plants that you're working from. But as you can see a little bit on the picture on the right, sometimes those hosts can really have extensive root systems. And so you don't want to just assume that whatever plant is growing next to the parasite is what it's actually parasitizing, right? It could be attached to a root from very long distance away. And here you can see some more examples of that, of these various specimens, most of them, I think, from Californian specimens. But that one on the lower right in particular, on this Eriogonum, which collected in Washington, you can again see how long sometimes that host parasite connection can be. So really cool natural history of these plants. Now, I won't show too many of these phylogenetic trees, but I think it's a good illustration of some of the evolutionary distinctiveness of these different lineages, even if they're very hard to tell apart, these different species. So if you're not familiar with reading these diagrams, they're really just like family trees of species, or in this case, each of the black tips with the text represents a different specimen that I extracted DNA from and sequenced for a variety of different genes for. And so I'll on the right-hand side, you can see some of those species names are in red boxes. Those are all some of the new species I'm going to be talking about for the next few minutes, and all of which are present here in Oregon. You can see a range map on the left. It's actually a slightly out-of-date range map, and I'll talk more about that later. But for the most part, you can see most of these species are all clustered in the western United States. The third thing I want to draw your attention to is the important role of the host again. And so each of these colored either green or blue bars or labels represent the hosts that each of those individual specimens was parasitizing. And so you can see a certain level of host specificity, not necessarily species specific, but oftentimes genus specific or a couple of related genera that turn out to be the hosts of those different lineages, those different species. So the first kind of new-ish species is Aphyllon purpureum. This had been recognized before as a subspecies occidentale of Aphyllon uniflorum. But it turns out that not only are they evolutionarily very distinct, as you can see from this phylogeny here, right? So the branches that connect Aphyllon uniflorum and Aphyllon purpureum are almost just as long as those that connect any of the other species. But also, there's areas where these two species come into contact, their ranges overlap in parts of the western United States, and you don't see any hybridization or anything like that. These are very genetically and ecologically distinct species. So Aphyllon purpureum is what is found here in Oregon, and compared to true Aphyllon uniflorum, will typically have more narrow, tapered calyx lobes, and often, but not always, the plant is smaller or just a little bit more. Seems to me a little bit more delicate than some of the eastern Aphyllon uniflorum. On the right, you can also see some range maps. I included both the global range of these two species and then also zoom in of Oregon. And you can see pretty much anywhere you go within the state, you can find the species, although it is a little bit less common in some of the more drier areas in the eastern part of the state, and also right along the coast on the far western part of the state. The old name Aphyllon uniflorum subspecies occidentale refers to west, right, the western subspecies. And for a long time, it was kind of thought, oh, Aphyllon uniflorum is in the east, and this occidentale, or now purpureum, is in the west. And some really interesting new research is showing that that's not totally accurate. So if you look at the range map along on the bottom, you'll see that there are some populations in Alaska, in the Alaskan Peninsula, of Aphyllon uniflorum. You'll see that throughout the Intermountain West, Montana, Wyoming, And also not shown on that map, but also in British Columbia, you'll see very small isolated pockets of Aphyllon uniflorum. As far as we know, it's not been identified in Oregon yet, the true Aphyllon uniflorum, but it is something I wouldn't be surprised if it were hiding out somewhere in on either Solidago or Symphyotrichum, in maybe some small habitat island, and I can, as it is in some of these other Western populations. These maps, by the way, are from iNaturalist. I'll talk a little bit more about that later, but a super great resource, both for the enthusiast, but also for a professional botanist or evolutionary biologist. It's been helpful in my research. Now, Aphyllon purpureum has a number of different hosts that are quite distinct that it can parasitize, and there's some limited evidence that these might represent separate host races, and maybe sometime in the future it might be worth naming them as separate varieties or subspecies, like this genetic evidence very weakly shows. These little numbers and the phylogeny above the branches refer to statistical support for how sure we can be that all of the specimens, group together as they do in those subgroupings. And in this type of statistical support, a 1 is the highest level that you can have. So these do seem to be very weakly genetically distinct and also very ecologically distinct on unique hosts. One lineage is on Apiaceae, right? So members of the carrot family. Another lineage, which is really common in the Cascade range, is on sedums or Saxifragaceae, which may seem like two very distinct types of plants, but they're actually, those hosts are closely related themselves. And then another group that's found on Antenarias or Senecio in kind of drier montane regions. It's also possible that this host race is on Coreopsis. There are some old collections from kind of near the Dalles, right in the Columbia River Gorge, potentially on Coreopsis that I haven't been able to get good genetic data from those yet, but I would suspect that they are in this kind of Asteraceae host race clade as well. So again, host specificity is really important here, and I haven't taken the next steps of actually giving these names because I can't find any other morphological features that can really effectively tell them apart. Every time I think I've seen something, right, see a distinction, I'll find some sort of counterexample. So these are very difficult to tell or impossible to tell apart if you don't know the host. But if you do, you can see that they do form these separate groups. So something to be aware of if you're ever seeing this Aphyllon purpureum in the field is make note of which of these three host groups it might be parasitizing. The next cool new discovery I'd like to talk about is this Aphyllon epigalium, and this was newly described in 2017 in the journal Madronio, but actually has a longer history of botanists noticing it as new. We found some herbarium specimens that pointed out its uniqueness back in as early as the 19 teens. Just annotations on the collection label saying, hey, these are kind of weird intermediates between a couple different species in this long pedicelled group. Then they're a different color as well. This bright lemony color is pretty unique in Aphyllon. And then more recently, it was actually described by Karen Cox who wrote her master's thesis in the 70s out of Chico State in California, but that name was never published validly. So, as far as the scientific community was concerned, she did all the work of identifying it as unique, but didn't quite, didn't quite publish it, and so we weren't able to use that name. And so, coupled with her master's thesis, and then additional genetic data and ecological data from Alison and I, we were able to not only describe this species as new, but also recognize two subspecies. One of which is much more widespread. That's the one that's here in Oregon, the subspecies Epigalium. And then a few isolated populations from Southern California that seem to be disjunct, the subspecies Nodocalifornicum. And again, I've shown that inset map of the different observations here in Oregon. The figure from our paper, the line drawing is only or almost only based on vouchered specimens. And you can again, you can kind of see that complement where our naturalists people have been able to observe these in areas that, couldn't be vouchered or weren't vouchered, and that complementarity of that data. As the name implies, these are found exclusively on the genus Galium or bed straws, in addition to their pretty unique morphological and coloration features. Again, that host really gives away its identification. So those of you who are interested, again, here's just a list of some of the defining features. It has these very long pedicels, usually 3 or 4 flowers per individual, and the host is always Galium and found in these forest understories. Finally, I'm just going to talk about this Aphelon franciscanum, which is a split from Aphelon fasciculatum. This is the key that is in the Jepson manual, the Jepson e-flora. The treatment, I don't think, in the Oregon Flora Project is out yet for this group, although, of course, you could correct me if I'm wrong. So I use that screenshot here. This is kind of the most recent update or taxonomic change in Aphelon. And it's a really cool example, again, like I said, of blending herbarium data with iNaturalist data. The challenge with this project is if we go back, I'm just going to go back to this phylogenetic tree, you can see that these two species are actually not even their closest relatives. So Aphelon fasciculatum is more closely related to Aphelon purpureum than it is to this other species that we were calling Aphelon fasciculatum. And so, great, we know that they're distinct, but it took a lot of work to try to figure out, again, how do you tell these apart in the field if you don't know what host they're on? Fortunately, I had a student who did a lot of work looking at iNaturalist observations and herbarium specimens. And through those two different data sources, was able to find a number of almost diagnostic features, right? So 2 or 3 different characters where if you put them all together, you can be sure that it's one or the other. Host is, again, like I said, always the easiest way to tell them apart if you can ascertain what that is, which is not always obvious if you're looking at the plant just growing above ground. However, the Aphelon fasciculatum is found on Artemisia species or sagebrush, and so generally speaking, if you're in those Artemisia, those kind of steppe environments or the sagebrush scrub, and you see the parasite, it's probably going to be Aphelon fasciculatum. Whereas there's other places, more closer to the coast, for example, that you would not have that host available, and then you can be sure that it's Aphelon franciscanum. This name Franciscanum comes from the Franciscan Formation, the geology around the San Francisco Bay Area, where the type specimen was collected. And this was a name that was actually already in the literature from another researcher, Daisy Akey, who wrote a taxonomic treatment of these species back in the 1930s. And again, she had noticed some differences among that weren't captured taxonomically. And she did some work into dividing up Aphelon fasciculatum into different subspecies. Didn't quite get everything right, but made some good progress on that. That then again we were able to continue to build on with these molecular techniques. So that's where that name comes from, even though it's a much more widespread species than just the San Francisco Bay Area or that Franciscan geology. Right, going all the way from northern Baja California up to the Rockies, and Sierra Nevada, all the way up to southern British Columbia, and eastward to the edge of the Rocky Mountains in Colorado and Wyoming. Aphelon fasciculatum has an even broader range, is from the Sierra Crest and eastward, or the more east of the Cascades, all the way to the Great Lakes region, and then all the way up into Alaska. So here's a few other morphological comparisons. Floral color was one of the big ones that my student Ben honed in on. Aphelon fasciculatum is generally going to be different shades of pink, sometimes very vibrant pink, like in this photo, but sometimes very pale, almost whitish coloration. Whereas Aphelon franciscanum is going to be more hues of yellow, sometimes getting as dark as purple or brown, but never pink like this. This is one of the features that that my student Ben really noticed by looking at these iNaturalist observations. And obviously that's not something that we would have been able to really see or appreciate just looking at herbarium specimens. Inflorescence shape or structure can be used as well. So the Aphelon fasciculatum have what are called corymbose inflorescences. So all of those flowers are all at about the same height. Right, so you can see how clustered those pink arrows are that point to the different heights of the flowers. You do have to ignore, sometimes there'll be a really young flower that's stunted or isn't able to grow to its full size, right? So you want to ignore that and just look at the rest of the fully mature ones. Whereas Aphelon franciscanum is verging on corymbose, but you can see that there's a lot more variety in how long those different pedicels are. And some flowers are a little bit longer, some are a little bit shorter. So we call it kind of subcorymbose or approaching corymbose. Another feature that to look at is the tip shape of the corolla. And so Aphelon franciscanum, again, will have more acute or pointed corolla lobe tips, whereas the Aphelon fasciculatum will have rounded corolla lobes. And finally, the calyx lobe shading. So if you notice those calyx tips, in this picture here, the tips of them are dark, and this really becomes accentuated when the specimen is dried, but you can often also see it on fresh specimens. Maybe 60 to 70% of the time will be dark tipped like this. Whereas Aphelon fasciculatum, you almost never see those dark tips, and it's just evenly toned. The base of the calyx and the lobes are all kind of the same hue. For those of you who are more quantitatively minded, there's a number of different features. These are all things that we determined by looking at herbarium specimens and taking a whole bunch of measurements and then using some statistical approaches to see which ones are more diagnostic or less diagnostic. And these, I already talked about the calyx lobe shading, but two of the most diagnostic ones are these quantitative measurements of the calyx cup to lobe ratio. So the Aphelon franciscanum seem to have a shallow calyx cup and much longer lobes, or a little bit longer lobes, versus the fasciculatum has a bigger cup and shorter lobes. And then also, sometimes this corolla is bent and often is more strongly curved forward in Aphelon fasciculatum than it is in Aphelon franciscanum, where the flowers are usually more upright and more point, towards vertical. And then finally, sometimes you'll just see these characteristics that, if you see this compression, this top to bottom compression of the corolla, it's Aphelon franciscanum. But if you don't see that, it could be either. So sometimes a feature is diagnostic, in one, if you see it, it's species X. If you don't see it, it could be species X or Y. And that's the case for a few different things. So I'll wrap up now. I want to make sure that there's enough time for Alison to talk more about the other species where there haven't been so many changes. But if there's any questions about my portion of the talk, I'd be happy to take those now.
All right, so I was gonna read off some of the questions in the chat. So the first question from Gail Baker is, Adam or Allison, can you address dispersal of parasitic plants? Do host and parasites have similar dispersal ranges?
Allison, I can't remember. Were you going to talk about that at all, or I'm happy to?
Well, you put your 2 cents in, and I'll do mine later.
Yeah, so that's a really great question. So these parasitic plants, first of all, have very small seeds, right? And each individual plant can produce tens of thousands, hundreds of thousands, or more seeds per individual. And so part of that makes dispersal of an individual seed pretty easy, right? They're small, they can easily be carried by water or even wind, perhaps. But the downside is, because they're so small, they don't have a lot of resources, and they have to find a host right away to be able to parasitize, right? Or else they're going to die. So it's not part of the question, but there's a whole fascinating set of studies that have looked at, how do these parasitic plant seeds find their hosts, and maybe we can talk more about that if people are interested. But in terms of the question about hosts and parasites having similar dispersal ranges, it really varies. I think a lot of times you'll have a host species that actually has a broader range than the parasitic plant species that parasitizes it. So, I'm just trying to think of a good example off the top of my head. But there's lots of places that you'll find potential hosts, and you could look all day long and not find the parasite. So the question is, well, why aren't they there? It's could it be just because they're dispersal limited over long ranges? Could it also be that there's very subtle differences in the biochemistry of some populations or not others that make it difficult or for parasitism to be unsuccessful. There are some interesting studies that suggest that I've been a part of that do suggest that there's been kind of parallel dispersal events of host and parasite lineages across from North America into South America of these aphyllon. So you kind of see the whole gamut in this group, but I think Alison probably has a few more things that you could add on that subject.
Yeah, I'll cover a little bit of that in my section.
Okay, so Tanya shares with us that Oregon Flora has not finished the treatment of aphyllon yet. So you're right, Adam. I guess Jepson is the most recent treatment that we have available to us. James asked, has there ever been an attempt to cultivate these or use them in restoration seeding? And it looks like Alison responded that she has been growing them, and she's going to talk about that coming up, aren't you, Alison? So just let's wait for a minute to see if she answers that question during her part of the talk. Tanya Harvey asks, how does one species evolve and diverge to use a new host? Ooh, that's a stumper.
Yeah, I can show a couple of other slides though that maybe shed some light on, well, that talk about the natural history of these host parasite connections. And it's really an open question to some extent, right? How we can't go back in time and watch speciation happen and all of that, but I think there's a lot of ways that the host and the parasite biologies are, one, closely linked, and two, there's a little bit of an evolutionary arms race, right? Between the parasite needs to figure out how to attack and parasitize the host. The host is going to come up with all sorts of defenses, and then can the parasite get around that and so on. So let me just pull up a couple of photos here that kind of illustrate that. Connection. Just have to fast forward here. And so I'm going to talk a little bit about the natural history of these, or the physiology of these parasites. If I can just share my screen, I've lost. And so what you're going to see are, think about the life stage of the host versus the of the parasite. And I mentioned how quickly this has to happen. And so it turns out that there's seedling cue, there's germination cues that the host is able to, that the host gives off these chemicals from the roots that the seedlings have to or can sense, or the seeds sense, and then the seedling germinates and grows up that chemical gradient towards the host. So that's kind of one level where you have this host parasite interaction. You can see this picture down below. These are 6-hour interval of a little seed that's germinating, and that little root tip, instead of growing down into the soil as most rootlets would, it actually grows towards the host root in whichever direction that host root is, and then forms this organ called the hostorium, like I said. And so you can kind of see that at the upper right. And then these are scanning electron micrographs showing how the parasite has to penetrate the host tissue first, and then form connections to the xylem and phloem, which are the transport tissues of the parasite. And so you can see this is a super intimate relationship between the host and the parasite. Even their cells on this picture in the middle, you can see the host on one side and the parasite on the other side. And these little dark channels are cytoplasm of the cells that can being shared. So they can just move stuff from one cell to the next. Whole chloroplasts, sugars, all sorts of things. And so, so the point here is that all these different stages, there are these defenses that the hosts have. And so if there's ever a shift onto a new host, it's going to be a very difficult, it's going to be a whole new set of defenses. And the plant, the parasite is going to need to adapt really, really quickly to that new environment or those new threats that could be, physical defenses that block the invading parasite cells into the tissue, could be a different chemical environment that makes it harder for those seeds to germinate or grow or attach, right? And so there's a lot, I could imagine a lot of natural selection on those new seeds to get them to, or on those parasites to really get them to specialize onto their new host. And then they become so specialized that you can't really hybridize anymore with your parental seeds, right, or parental lineage that's on the very different host, because if you hybridized and mixed those genes, you wouldn't be able to, parasitize maybe either one. So that's kind of my working hypothesis. Kind of a, I don't know how you necessarily go about testing that in a general sense, but you certainly see the pattern of these very host-specific lineages of parasites, which I think is really consistent with a kind of a host ecology-based approach or way of thinking about that evolution. Just as you see, plants endemic to different types of soils, right? To serpentine or to, gypsum dunes or to, all these unique geologies. Well, for these parasitic plants, their host is just like their unique substrate. So those are my 2 cents.
Do you want to add anything to that, Allison? No. Okay. One final question while Adam's still here. Everybody, Adam's 2 hours ahead of us, so he may book at the end of his presentation. Linda asks, I'm interested in learning if we have annual species such as Rhinanthus minor. Do you guys, can you, either of you answer that question?
I'll say that some of these parasites, like Aphyllon purpureum, are annuals. And I don't know what hemiparasitic annuals there are in Oregon. Rhinanthus is definitely around in the Western U.S. I believe I used to see it in Washington State, but I don't know if it's in Oregon.
Okay, we'll follow up on that one. So, Allison, I think you can go if Adam will give you the screen.
Yeah, Adam, if you unshare, I can share. Okay.
All right. And
Thanks,
I can stick around a little bit longer. So if there's questions after Allison's portion that I can help answer, or people think of additional things, I'll stick around for a little bit yet.
Awesome. Thank you.
Okay. Can you all see this image?
Yes, we can.
4 panels. Okay. So this is just to summarize again what Adam had started to tell everybody about this group, Aphyllon. And he talked about what was formerly Orobanchaceae section gymnocaulus, the naked stem, in honor of these superlatively long pedicels that the flowers, they're like little submarine periscopes coming up out of the ground. And they actually do have a little stem with axillary, like little bracts that probably represent their former leaves, but they're very reduced to just little clasping bracts, and then axillary flowers. And but the whole stem part is basically underground. So this is Aphyllon purpureum, and it's was considered to be in a section. So Aphyllon, I think, was named, as Adam said, named by Asa Gray for some material that John Clayton, a preacher in the colony of Virginia, sent to him. And that name persisted off and on. And then when this whole group of holoparasites got monographed by Beck von Managatea in 1930s, he was European, and he had a synthetic view and said, wow, these things look so much alike. They're all going into one genus. And there were 4 sections. He may have recognized more than 4 sections, but there was section Gymnocaulus. And then the other section in the New World was, he had recognized as section Mycorrhiza. And that one has above ground inflorescence stalk with flowers on pedicels. So it's basically a raceme above ground. And then 2 other sections of Beck's that are important to you Oregonians. One was one that became the genus Phylogenetic. That was, you knew it as Orobanchaceae ramosa. It's been a crop pest in the United States since the days of the colonies, because it was on hemp back in the day when everybody grew hemp for rope and clothing. And it's also a big problem on tobacco and tomato. And here in California, it's a very big deal on tomato crops. And there's a lot of research effort trying to figure out how to limit it since it's here. As far as I know, it's not in Oregon yet, but you should be on the lookout for it. So that genus Phylogenetic has what we call bracteoles. They're 2 little tiny, I don't think we can see in this picture, but I'll show you some later. There's little tiny bracts, like little whiskers on either side of each flower. And section Mycorrhiza also has that, but section Gymnocaulus does not. And also the 4th section that Beck had called Asproleon, but that is still Orobanchaceae. And you have this nasty one in Oregon. It's Orobanchaceae minor, and it's on carrots and alfalfa around the Portland area, and is a major agricultural headache. So I've listed a couple of field traits, their presence of absence of bracteoles, long pedicel, short pedicel. The old world pests have no pedicel. The flowers are absolutely stuck onto that stem. They also have a split calyx, so it's got a split top and bottom, so it's like two-parted. And the other interesting thing about these, and I think this is why they are good crop pests, is that they're determinate in the sense that you can see in these pictures, there's a few buds left at the top, but these things come bombing out of the ground when they're mature. They flower pretty much all at once up and down the stalk, or very close succession, and then they set seed with or without a pollinator, and they're done. So they can complete their life cycle within the space of time that a crop is in the ground, basically. Whereas our New World plants are more indeterminate in that they will open a few flowers, and then if the rains don't come, they'll just sit there, and then open a few more flowers later on when there's good weather, or they might even, if it gets really hot and dry, the upper inflorescence will die, and then branches of the inflorescence will form in the lower axils. So they're more leisurely, and although the aphyllons in section mycorrhiza, like down in the desert, have been known to parasitize, to come up in tomato fields on occasion and tobacco fields on occasion. It's when the field has been planted into their habitat and they don't really make it because they're a little bit too slow. So let's see. So here's a picture of Orobanchaceae minor. As you can see from this iNaturalist map, there's a hot spot around Portland, and here it is. It's in all its purple hairy glory coming up in a field. You can see the clover right next to it. And these things produce tens of thousands of seed. They will saturate the soil with like a cloud of a seed bank. And so any host plant, such as alfalfa, that germinates and its roots grow through that cloud, very likely the root will come within a few millimeters of a lucky Orobanchaceae seed and therefore get parasitized. So here's Phylogenetic ramosa, and my colleague Pershing Hosseini here at UC Davis, she's got scrubs on because she's in a biological containment level 3 lab where she's growing this crop pest, Phylogenetic ramosa, as well as Phylogenetic aegyptiaca, which is a relative that just showed up in California about 10 and 15 years ago. So they're both on tomato fields in this area. They're under a great deal of study and attempts at containment, and so far that has not happened, but they don't seem to have spread very far either. And you can see her kind of cool growing chambers. They're they're semicircle pots with tomatoes in them, and they have a plexiglass window that is kept covered with plywood until she wants to study the development. And she's testing different cultivation conditions and different herbicide regimes to try to rid fields infested with these. So those are two of the groups of Orobanchaceae and Phylogenecae. And one of Adam and our motivation for moving the New World plants back out of the Orobanchaceae group was in part because they were all on noxious weed lists across the US, with basically a list, kill on site. And a lot of our New World taxa are rare. And so we didn't necessarily want them to be fall victim to any overzealous eradication programs. So here, one of you asked about cultivation. I've been growing these in pots for decades now. What I do is I get a little host plant going in like a 4-inch container, and then I pot it up into a 1 or 2 gallon container, and then on a pad of soil at the bottom, and then I mix the aphyllon seed with the potting soil and tuck it in a sort of donut around the edge of the host, and then I put some potting soil on top. And so what this image shows is a host test. And what I'm testing here, this is Aphyllon uniflorum from the East Coast. I collected this near my sister's house in Halifax, Nova Scotia. And what I've been testing it on, this is on the picture on the left is last June. It came up on its native host, which is Solidago rugosa. I collected seed of the host at the same time and grew it out and then planted the aphyllon on it. And then on the right is Solidago velutina from California. And so it grew very happily on both East Coast and West Coast Solidagos. So it's anyway, that's a useful piece of information. What the plant would not grow on is the Eurybia divaricata, which is an aster relative that it's been reported on up and down the East Coast. So either that's a false report of a host, or there are two cryptic aphyllons with different host preferences. So we're going to have to get a hold of some more seed and do a little more testing. And the picture on the right is, this was this morning actually, this same pot in the Solidago velutina. This year I unpotted it this morning to see if anything was going on, and sure enough, there's you can see little aphyllon plants starting to make their way up to the soil surface. And you can see this little sort of star-like cluster of their fleshy little roots at the bottom. And there's actually quite a lot of I see a whole bunch of little clusters of these plants. So interestingly, the seed made its way all the way down to the bottom of the pot, where its hosts are root bound, going round and round and round the bottom of the pot. So very likely to have grown past an aphyllon seed. And then the inset at the bottom was the little plants in the soil in Halifax in the wild. And you can see that some of them these are maybe a millimeter or a centimeter tall, and that's a host root coming in, and the little plant's attached to it, and it's just growing, and once it gets big enough, it will flower. So these were in the soil around the flowering plants, and I think those may be seedlings, and they'll be big enough to flower next year. And the seedling at the bottom you can see more of its roots. They're kind of thick and fleshy and kind of bifurcating in root growth, but they're very stumpy. So they're really more, I think, maybe they do have this species has secondary host attachments, but so it makes haustoria on multiple little stubs. But I think those roots are probably partly just starch storage, getting ready for its quick growth when it starts to flower. So I was asked by Gail to give a little romp through Oregon taxa, ones you might see. And so here we go. This is Aphyllon pinorum, and its host is Holodiscus discolor, not pinus. I guess it does grow in pine woodlands, so maybe it was named in honor of the habitat, not the host. We'll give whoever named it the benefit of that doubt. And this one's interesting because it's actually a short-lived perennial. And you can see in the lower left, that's a dug up one in my hand, and I'm holding its inflorescence out of the field of view, but you can see a couple of aborted flower buds. And then that sort of rotten part at the right is the last year's inflorescence, which had rotted off and died, and leaving this kind of like fleshy, what the taxonomists in this group call a tubercle. And it was attached to a host root, which I, in my impatience to dig it up, broke off. The host-parasite connection is very fragile. So this plant, Aphyllon pinorum, the yellow line is its range. It's all the way up through Idaho and British Columbia and Washington and down into the North Coast ranges and the northern Sierra Nevada, and it has a disjunct population on Mount Lemmon in New Mexico, and there's one Sky Island instance of it near Elko, Nevada, on the Holodiscus discolor subspecies microphyllum at 10,000 feet. So the theme for these plants is they parasitize widespread hosts, but you never see them everywhere the host is. And so that's that was under discussion a few minutes ago, like how do they disperse? I think that they are basically in very stable habitats where host plants have been there for a long time and have plenty of water so that they don't triage their root systems and cut off their little parasitic friends. So a very wide-ranging species. Another wide-ranging species is Aphyllon corymbosum, and you will see this in the arid areas of the eastern part of the state. In the upper left is Vantage, Washington. You can see the Columbia River in the background, and you can see a couple of little like brown tufts right there next to a couple meters from their host, the big sage, Artemisia tridentata. And one of these two tufts I dug out, and that's here with the dime for size. And you can see that this one has a big fleshy underground stem, a few little pathetic gestures in the direction of root system. And then its above ground part is about 4 inches to 6 inches tall. And so corymbosum, like the habitat and the host, like it's always on Artemisia tridentata, and it has these reddish sort of what I call Mickey Mouse ear look to the upper corolla lobes. And then it has the lower picture is a little bit sort of faded, but it has this humpback corolla, kind of reminds me of like the back of male salmon when they're spawning, this big hump. And what's kind of a problem I've been grappling with is that 2 subspecies of this one have been described based on the inflorescence shape. So there's Mutabilis has got this sort of slightly erect raceme, and then the traditional, if you will, corymbosum has just a few flowers in not a true corymb, but it's a very flat-topped inflorescence where the flowers are close to the ground. And these things are so variable and individualistic. I'm not really convinced that it's not just like a local sort of like nutrition status thing. It's something that we really need to look into more. So, but moving on, I'll leave you with the corymbosum problem and introduce you to another one. Here we have pictures of Aphyllon californicum, and there's 2 subspecies that are in Oregon. The one, well, actually not in Oregon. The one at the right is Californicum, subspecies Californicum. This was first found by David Douglas when he was making hikes out of Fort Vancouver, and he found it in, he found a Aphyllon in the camas fields that were, he mentioned that they were meadows that the local tribes people were burning, and they were probably camas fields. And so he found actually the one at the left, Grayanum, and there's a picture here. This is actually a meadow in the Sierra Nevada, and the inset is a picture of Aphyllon Grayanum in one of its favorite hosts, Aster or Rigeron, and it goes on any number of species of those 2 genera. But it is closely related to this Aphyllon californicum that this picture is taken in Humboldt at the Big Lagoon area, and it's where it loves to be on Grandelia and the Strand. You can see that, like, kind of that high tide detritus there and rounded rocks and sand. And it also is all over the place in Puget Sound, but for some reason, it's not been reported on the coast of Oregon, and I don't really know why. I know that there's been some pretty pervasive disasters on the coast of Oregon with like the buried tree trunks, meaning like there's been subsidence and other sort of things. So maybe it's there and you might find it, keep your eyes peeled, or maybe it was eliminated in some, earthquake disaster in millennia ago. So the thing I want to tell you about the Grayanum is that this one was thought to be, I think I actually have a range map. The Grayanum in Oregon was thought to be extirpated for many years, but it's been popping up on iNaturalist in the Willamette Valley. So this one you should be able to find. It's easily overlooked because it's in dense vegetation. And of course, it's also ephemeral. It'll be up for a week or so in July, and then it'll turn brown and quickly deliquesce and set its seed. And so you have to be a bit eagle-eyed. So this is something to look for in vernally wet meadows on Aster Origeron. So the Aphyllon californica in the yellow, you can see that it's up in Humboldt County, and also in Puget Sound, but mysteriously has not been reported in the intervening coasts. So Aphyllon californicum grayanum is also in wet meadows in California, and Adam and I were startled to discover that it's not only parasitizing Aster and Origeron, but also Euthymia occidentalis, which is a late summer blooming Asteraceae of sort of marshy riverbanks. And I bet it's been hiding in there all along, because what person in their right mind would wade through muddy riverbanks in 4-foot tall Euthymia looking for parasitic plants? So Aphyllon ludovicianum, this one is a kind of a hallmark species of the Great Plains and the Rockies. It was first discovered at Fort Mandan by Lewis and Clark and sent back east. It was in the dry plains. It's this little itty bitty thing on various Asteraceae, Artemisias, Chrysothemis. That's the palm of my hand. So that whole plant, that's from the Platte River area. It's only about maybe 5-6 centimeters tall. But in the western U.S., in the Columbia River Valley specifically, there's a subspecies, Arenosum, that gets much taller, almost a foot tall in some cases. And where you can see that is on sandy areas along the riverbanks. So it's on Dalesport Road. It's at the end of the runway of the airport in the Dales. So you can go there and see it in the, there'll be the people watching the airplanes take off, and then there'll be you looking for aphyllons on Chrysopsis villosa. You have to park where it says, do not under any circumstances park here. Or at least you did back when. I don't know what it's like. I haven't been there in a while. So one of the things about this ludovicianum is a very polymorphic, lots of different hosts, lots of different lineages, and we're starting to kind of peel off different ones. And several years ago, Turner Collins, George Iskevich, and I described Aphyllon riparium, and that one is a tall plant. Its hosts are Ambrosia trifida and Xanthium strumarium, and it lives on riverbanks, and you can see it's like the sandy deposits on riverbanks. I have an illustration. So here's the kind of Great Plains, a vanilla flavored Aphyllon ludovicianum with the Arenosum that is kind of specialized on Chrysopsis in the Columbia River Valley. And then the black squiggles are the various rivers where the Aphyllon riparium has been found. Once we described it, people started finding it in more and more places. So it's on the Potomac River and the Rappahannock in Virginia. It's on the Ohio River. It gets all the way out on both forks of the Platte River in the west. So yeah, you may see Aphyllon lutevacianum on aeolian dune fields in the Great Basin and Great Plains area. Any place sandy that has a good Asteraceae host. So just one more cool holoparasite I had to add in here. In Oregon, you have Copsiopsis hookeri. It used to be Bosniakia hookeri, but we saw fit to split them out based on DNA sequence information and a whole lot of different morphological characters. You can see the Alaskan, this one's actually a true Pacific Rim taxon, Bosniakia rassica. It's on alder. It blooms in the coastal Alaska and Queens Charlotte Islands areas in June. It's a very elongated inflorescence, whereas the hookeri and our Copsiopsis strobelacea are very tight little conical structures and somewhat different floral morphology as well. So the Copsiopsis hookeri is on Gaultheria chalone, although when I lived in Seattle, Ravenna Park in Seattle, it was on arbutus, on a madrone there. So there's some question about these two plants in terms of they have very similar hosts. There's been discussion about in Northern California near Trinity County, Happy Camp area, that there seems to be some integration of traits. So anyway, it's up for debate what they represent in terms of, are they a grade, a latitudinal, different manifestations of a latitudinal gradient, or whether they're distinct. Nobody's tried to grow these. They're slow growing perennials. They probably don't flower for several years, and trying to get them going on a manzanita, I don't know, like that. It would be a fun exercise, but you'd have to be rather patient. So here's a picture from Cedar Knob, Washington, when I lived in Seattle. This was just an hour or so east. There's a huge population of Gaultheria chalone. Actually, most of the eastern Washington is a big population of Gaultheria chalone. But what's interesting about this population of Copsiopsis is that there were yellow individuals, purple individuals, and individuals that were sectoring purple and yellow, making me wonder if there was a transposable element or something causing that color swapping. So there can be both colors, they can be in the same population. So that kind of throws people off, but it's just the way it is. They like to mix it up a bit. This is a scanning electron micrograph of a seed, and you can see so Copsiopsis has relatively enormous seeds. These are they're like a millimeter or more. The seed, the embryo inside is very tiny. It's less than 1/10 the length of the seed. So the surface is all this sort of spongy tissue, like dead tissue. And the thing about this is, it does two things for the seed. One is it traps air bubbles so that the seed will float very nicely. It also greatly increases the size of the seed without increasing the weight. So it would also be airborne very easily. So in terms of dispersal, these these plants live under the canopy of Gaultheria chalone, so I doubt that they're blowing that far in the wind, but they probably are floating off in with heavy rains. And what would be nice for them is that they get washed downhill, they get buried in a bunch of organic matter, and where else do roots love to go than to explore the nutrients in piles of organic matter, which is just where the parasites are hoping the roots will come. So I think in terms of dispersal, yeah, they're probably not great dispersers. These plants are so close to the ground, but they do have this sort of spongy surface texture that gives them an advantage under inclement weather. So that's my last slide. This is a banner day in Yosemite National Park when I worked there, and we went at Karen Amstutz, the gal with the long curly hair and binoculars. She's the ranger naturalist for Tuolumne Meadows every summer, and she had hiked up Mammoth Mountain that's on the south side of Tuolumne Meadows and found aphiloms all over the place up there. So we got a little group together with Adam on the left when he was a grad student, and Garrett Dickman, who is another one of my colleagues in the park, and we hiked up and found hundreds and hundreds of them on Artemisia vaziana. So it's up at the highest levels of the Sierra Nevada. And with that, I will take questions. Let's see. Stop share.
Okay, well, Doug asked, could you describe some crop impacts some more? And Adam responded about the native species not having that many impacts to crops, but the invasive ones are destructive and pathogenic. I think maybe Doug was asking a little bit more about the invasive ones in the Portland area.
Yeah, well, Orobanchaceae minor, it's been there for decades and probably came into Oregon on contaminated seed. The seed being very small, it could be easily undetected. And when I lived up there, and that's now 20 years ago, there were certain counties that were quarantined and that they could grow I guess carrots is a big crop in the Portland area, or was, and the farmers could grow carrots, but they could not sell carrot seed, or they couldn't market carrot seed. So it seems like it's persisted there. So I would guess that the quarantine area for not allowing selling of crop seed has gotten larger. It's also in southern Washington in the battleground area. And there it's gotten into alfalfa fields or meadows that were formerly cultivated alfalfa. And there's an awful lot of clover around, so eradicating it is probably hopeless.
Okay, thanks. Let's see, James mentioned that he had found a Phyllum californicum on a Grandalia, which down here in an area that in the Willamette Valley. It's probably not a big surprise as it's an aster, right?
Yeah, so Aphyllon californicums are, there's several subspecies in California that aren't in Oregon, and they love their Asteraceae, and that's surprising because a Fern Ridge RNA near Eugene, so that's well inland. So then the question is it the gray anum that's decided, okay, it'll go on to Grandelia, or is it the coastal subspecies inland? And so it could be either one. And that's a really interesting observation, and I'd sure like to know more.
All right, there you go, James. You've got your work cut out for you. Maybe you can put something in iNaturalist for Allison to see. All right, we got one last question from Tanya. She says, Castanea also have that mesh covering on their seeds. Do aphilin's and other members of Orobanchaceae also have similar seeds? You mentioned,
That reticulate sort of spongy coating, and it has been speculated that it acts as a trap for the host root, like the host root would grow through that and then it sort of locks the seed onto the host root. But it's clearly advantageous in some way because pretty much all of the parasitic plants have it in the New World. Actually, I think the older ones do too. So the only one who doesn't is very interesting. It's the one I did my PhD thesis on, Conophilus americana, and that's a perennial. It looks like Bosniakia and Copsiopsis, but it's a parasite on red oaks in the eastern U.S. And that one has the spongy layer, but it's got a smooth outer coat. So it's a shiny round seed, maybe a millimeter diameter. And the interesting thing about that genus is that the seed capsules don't open, they're not bivalved. They're indehiscent, like they just don't open. But if you go to a population in the summer or fall, when the little inflorescence stalks are all dried down, you'll see a neat little hole drilled in the bottom of each capsule. And if you catch it just at the right time, you'll see a line of ants making off with all of the seeds. And they have a little eliosome on them, a little oil body that the ants are. So the ants are going off to carrying these seeds off and basically planting them someplace near the oaks.
Near their nests.
Not every single one of those parasites has that reticulate surface seed coat.
Great. Well, thank you very much. I think if everybody wants to turn their video on, and let's give our speakers a warm thank you for.
Thanks, everybody.
Hi, Alison.
Hey, John. Hi, Tanya. Hi, Rex.
Thank you so much.
I hope it inspires everybody to go out and look for them and report them. Adam and I are looking on iNaturalist for new finds all the time. So you
Excellent.
can also email us.
All right.
All right.
Thanks,
Thank
everybody.
you so much, both of you.
Hi, Rebecca.
I just put my email in the chat. It's aschneider2